Okay, good morning, everyone. We're going to get started. So today is the last lecture of the semester. And so we're going to combine everything that we've learned so far, from the bacteria to our human immune defense mechanism, and to look at how bacteria are trying to infect us, and how we can hold the bacteria in bay, preventing them from infecting us. And so we will use all the knowledge that we've learned through the semester and to apply to specific examples. Now, typically there will be a class called medical microbiology follow up with this particular class, and a virology class that's running right now in the spring. Because usually this class is runs in the fall. So this builds up. So since we only have one lecture to talk about infectious microbes, and we will only focus on a few well-known examples, and so hopefully you can then apply the same principle to other microbes. So we'll focus on airborne microbial disease, and direct contact microbial disease, and sexually transmitted microbial disease. Okay, so here's a list or a graph about mortality per 100,000 to graphing the death due to the infectious disease in the last century, actually. So from 1900 to 2000, right? And then you see, as we're getting better and better, the mortality rate caused by microbial infection went down dramatically, except there was one peak period in the looks like 1917, 1918. Does anyone want to guess what happened during? I mean, this is real. This spike was real, but why? Does anyone want to take a guess? Why do we have such a high mortality in the 1917 to 1919, 1918? Anyone want to guess? Put in the chat. What happened? Yeah, one possibility. Anything else? And there's also World War One. Remember, trench warfare, and that actually contributed to a lot because soldiers were wounded, and they are in the battlefield. They can be tended, so a lot of people died due to microbial infection. So we're going to break up into three specific areas. So here are the microbes or the disease that we'll be talking about individually. Okay, so we're going to first talk about this picture, the sneeze. By now you've seen this particular year. Well, usually when I give this lecture, I mean, this will be the first time anyone sees sneeze. But I'm sure this year is different, and you've seen lots of pictures like this, right? And these are the droplets, and I'm hoping that you're convinced that this guy, if anything that's up in his nose, it's going to come out here. So whatever there, the microbe, the COVID-19, the virus, see everything is going to be here. And this is within the arm length. And now, because of the pandemic, and then there are lots of actual studies using lasers, and you probably have seen them, to track how big or how small the particles that can fly. And so majority, when you sneeze, majority of the particle is about 5 micron. They come down. Okay, like more than 5 micron. But if it is larger, smaller than 5 micrometer, they call micron, and this is the size, and this is compared to your hair. Okay, human hair. I guess it varies between 60 to 100 micrometer, right? And so that's the droplet. And so obviously the smaller is there, and it will float in the air longer. Um, and the in terms of virus, then if you have multiple virus forms a core, and that will stay as a particle and then transmitted through. And the effect is more problematic for human lung. And as we said last week and before, that our lung is really not protected by anything strong. We basically rely on the mucous membrane to trap all the particles that's going in. And so here you have telling you about the velocity, how fast the air traveling through your lung. And this is actually very, very fast. You know, this is measured by centimeter, so 150 centimeter per second. Okay. And then this side is the particle size, tells you the particle size. The bigger the particle gets trapped, right? So 20 micron gets in here, but if anything less than 3 micron, and that will actually have a chance that getting through all the traps and then to the lung itself. Okay, so and so now you've throughout the semester you've looked at, or well, you've seen pictures of different microbes, right? And so these are the smaller ones, and these are the smallest, and then they tend to go by the size, and the smaller they are, the more likely they're going to get to the alveoli than the where the air exchange takes place, where there is actually no protection. Okay, because you need air and efficient exchange. So at the end of these respiratory tract, there is no protection, right? So if bacteria goes through, then that's a problem. So that's why it's more important that the density is important. Imagine if you were right in the middle of this guy, and if your nose is here and you breathe in, say, oh yeah, well, okay. So these particles, right? They will such dense area, and majority will be filtered or captured by the lung, but a few will get in. Knowing how fast the bacteria can grow, only one bacteria gets into your lung, that will effectively cause an infection, right? So at this point, I want to pause and ask, how fast is E. coli growing? What's the generation time, E. coli? Anyone want to type into the chat? Anyone want to be the first person to type in the chat? E. coli, how fast E. coli grow by generation time? Well, yes, 30 minutes. Okay, 30 minutes under best growth condition. What's the best growth condition? 37-degree and lots of nutrient. What's the temperature in your lawn? 37-degree. Lots of nutrient. So if E. coli gets in, yes, it will cause an infection. Okay. Fortunately, most of the time we can avoid that. Okay. So so these are the pathogens that can cause infections. And famously, Staphylococcus aureus that can cause upper respiratory infection, and Streptococcus pneumoniae can cause the lower respiratory tract infection. Then you have pertussis, Bordetella pertussis. So these are the bacteria, and then you have chlamydia, Cladodrophila. I'm sorry, they cause lower respiratory tract infection. And so we're going to try to one particular streptococcal infection. So Streptococcus pneumoniae, so this is a picture of Streptococcus, right? They form long chain. Okay, this is different from Staphylococcus, right? Streptococcus forms long chain. And if you stain them under the microscope, this is what you see. However, if you use Indian ink to do negative stain, and this is a picture of negative stain, what would you see? This is white area. What do we call this? Anyone want to venture? When you see, so first of all, what's a negative stain? A negative stain, well, we know staining is to get a dye into bacteria and then make it either fluoresce or show some color, right? You see gram staining, and so the bacteria becomes red or pink or purple, right? But negative stain is like big giant particles that you put it on to the slide. And so then the dye particle are so big, it wouldn't go through a layer of, you want to guess before I say it? Yes, capsule. Thank you, Hala. So it's the capsule. Remember, they have a capsule. So now you can see how thick the capsule is, right? Now, if you bring back what we know before, we talked about this particular capsule before. So if the Pseudomonas produces capsule, it's likely to form smooth colonies, right? Because these capsules combined, they basically form a slime layer. Now, why is this important then, this capsule? Well, because think about what do we rely on to kill bacteria? One of the things last week we talked about to kill bacteria is called, starts with C. Anyone recall? that starts with a C, describe a whole series of protein that they can actually punch a hole on the surface of bacteria, right? That's called complement. Now, to activate the complement, what do we need? The complement is a set of protein flows around. It only is activated when the complement 1, the C1 protein, gets trapped to two antibodies attached to the surface of bacteria. Now, if you have antibodies, will they be able to get to the bacteria that's in the middle? No, right? Because antibody will be here. It can't penetrate. So now this bacteria, can it be killed by complement? It can, because it can't get to it. Can this bacteria be captured by phagocytosis, by the macrophage, or even activated macrophage? It's very hard because it has this slimy layer on the top, and the macrophage grabs it, slips away. Right? And so this picture actually shows you a lot about this bacteria and explains a lot why this bacteria can cause an infection. And it's a very bad infection because it's difficult to kill this bacteria. And that's why this capsule is a virulence factor, right? So allow the bacteria to kill us. And so this is one example of how bacteria is producing virulence factors to kill it, to avoid to overcome our immune defense and then establish and cause an infection. Okay, now on this we will have to rely on antibiotics to kill the bacteria. However, the numbers of antibiotic-resistant bacteria is really on the rise, right, in the hospitals and all around. And so that's is predicted to be the next crisis. As a microbiologist, I pay more attention to that. And I'm also wanted to plug in a small advertisement for my lab. And so we are actually studying the how can we generate, how can we find new ways to kill bacteria? And we have an active project in the lab trying to figure out what can we do to kill these bacteria. And we actually make some progress. We actually have something that can actually kill the bacteria. So if any of you are seriously interested, wanted to do, have time to do some bench work, to do research, and I would encourage you to contact me, and we can talk. I can explain more. All right. Okay, so that's an example of a Streptococcus pneumoniae infection. By looking at this bacteria, we can visually see this virulence factor and how they can cause an infection. Another bacteria in the family, Streptococcus pyogenes, can also cause what's called a scarlet fever. And this bacteria produces a toxin. And what's also and this toxin itself causes the whole body reaction. And you see, the toxin causes the body to react to produce this entirely red rash. Okay, this is a sign of the infection. And what's also interesting about is this bacteria doesn't always cause this kind of infection. It's only when this bacteria is infected by a phage. And a bacteriophage lysogenized or incorporated into the genome of this bacteria. And then, so now this bacteria is producing this toxin. So this is an actual example of how lysogen. Remember lysogen? When we talked about phage, we talked about bacteriophage incorporate themselves into the bacteria genome and then being carried on. And so this is an example how this toxin can actually, encoded by a virus, can actually cause a infection and toxic reaction. And so these are the streptococcal infection, two examples. And so we can use blood agar to diagnose to see if this is a toxin, if this is which streptococcus that you have. Now in the lab, you probably did a blood agar exercise trying to see hemolysin, whether hemolysin is produced. And a streptococcus that produces hemolysin is most often more pathogenic. And we can also use what's called the ELISA assay. That's an antibody mediated detection tool to detect whether that's a streptococcus infection. The treatment is relatively easy. We can use penicillin to control the bacteria, to kill the bacteria, or to prevent the infection. Now, at this point, I wanted to pause and ask, what does penicillin do? Does anyone remember? Which biological reaction does penicillin block? Not specific enough because I'm asking which biological reaction it is blocking. Keep going, keep going. Someone volunteers. Um, yes, cross linkage of close. Just one more thing to say. What's cross linking? What's being cross linked? Peptidoglycan, yes. The site, yes, the bridge, the side chain. Excellent. Thank you. Okay, so that if you look at this series of questions and then you understand when people ask you, so what biochemical reaction? It is the cross-linking of peptidoglycan that this penicillin is blocking. Okay, and so then you can say, okay, so if someone asks, okay, what's the cellular structure this penicillin is targeting? Then you can say cell wall. You see, cell wall is a cellular bacteria structure. Okay, so that's different. So it's about chemistry and the actual cell structure. Okay, so and then we talked about later on, we talked about penicillin and how penicillin can be spread and how penicillin can resistant bacteria how does bacteria resist the penicillin, right? We talked about the enzymes that can be produced by bacteria that actually break up the consensus ring, okay, beta-lactamase ring, and that's an enzyme called beta-lactamase, right? And so that's the penicillin. So everything is actually, this slide looks really simple, but this is kind of like a review sheet. Tells you that, well, these are the things that you need to know, okay? And we can also, people could also ask you, oh, okay, so penicillin, how does penicillin kill bacteria and what's the effect? Well, it breaks up, it prevents the cross-linking, so the cell wall, well, then let me step back. So it depends on the people, right, who's asking the question. If a non-biologist asks you this question, then you say, okay, yes, penicillin basically will weaken the bacterial cell wall so that the bacteria will grow and the cell wall will break up and bacteria will break up and die. But if it's a microbiologist or bacteriologist asking this question, then, or like me, I'm asking, how does penicillin kill bacteria? Well, then, yes, you need to be more specific and say, well, it prevents the cross-linking of the peptidoglycan, so the peptidoglycan is weakened, and so when bacteria grow, the peptidoglycan cell wall simply just bursts and bacteria is killed. All right? Okay. So the next disease we want to talk about is called tuberculosis. And this is caused by mycobacteria tuberculosis. Okay, and the disease is caused by this bacteria, and early on we talked about this bacteria. Anyone remember what's unique about this particular bacteria? It's one of the gram-positive bacteria. It has a waxy layer. Yes, the mycolic acid. Perfect. It has a mycolic acid layer on the outside, and we said it's really tough. It's preventing everything. It's like putting a layer of wax outside the bacteria. Now, what's the effect of that? Well, if you have a wax on the top, on the outside, then would it be able to kill by complement? No, because antibody can't even bind to it. Can it be captured by macrophage? And not very effectively. Can it be opsonized by antibody? No, no, no. Okay. So this bacteria, if it gets into your lung, it will be able to establish and then grow and then infect the lung tissue when you breathe in, right? And then your body will react and send macrophage capture the bacteria. No, it can't. Even if it captures the bacteria, it still cannot lyse the bacteria. Remember, macrophage capture the bacteria relies on what to kill the bacteria? Reactive oxygen, right? Hydrogen peroxide. Well, this bacteria is completely resistant. It's like they have a whole shield, right? They have an entire steel shield, and so it doesn't really matter what you're shooting at it. It's just not going to kill it. And so what then happens is one macrophage captures it, cannot kill it because this bacteria just keep growing inside the macrophage until the macrophage die. and then the next macrophage comes in and trying to kill it. And so your body essentially put together a whole layer of macrophage surrounding it. And so it becomes a spherical, or we call it tubercle. And so inside this, then you basically have a liquefied, have a liquid with bacteria in the middle, with with tissue formed by macrophage on the outside. Okay, and the tissues are so dense, and they are visualized. They can be visualized by X-ray. Okay, and so this is a normal X-ray of a person. I often ask people in my class, do we know how to read the X-ray? I know some of you, half of you want to go to PA school, half of you want to go to medical school and dental school and all that. Do we know how to read X-ray? The white area represents, anyone want to type in? Respond to the chat. What's the white area represent, or black area represent? So the white area represent ribs. Okay, good. Can I ask why rib is white? Does anyone know why rib is white in this X-ray photograph? Yeah, calcium and bone density. Yes, the bone is very dense, but why bone dense is white? Okay. Um, well, hmm. Very nice. Very nice. Yes, the dense bone blocks the X-ray passing through. Okay. And sorry. Can I ask why then, yes, blocked X-ray passing through, and so why is it white? It has to do with the chemistry. And so this X-ray, these are, and you guys are very lucky, born in the age of color film. All right. Right. I was born in the age of black and white. Um, and so the black and white film relies on a very interesting chemical reaction. It's a silver. So and so basically the silver, when it's a silver chemical, when it's react to the light, and then it will it will form silver, and then uh, and then becomes and so then it will stay on the film. And so now you can see the dense, the amount of the X-ray that passing through the bone can be detected by how much silver reaction is happening. And because your bone is so dense, it blocks the X-ray from passing, so less silver is converted. And so versus the tissue is soft and is transparent to the X-ray. Okay. Now that's enough about the a little break, right? Sidetrack. Okay, now the reason we're talking about this is because we earlier I said there's a tissue and that's so dense that's visible in X-ray. So now if you look at this person, and this person, you see the rib, you can still see a little bit of rib, but notice in contrast how white these areas are. Notice these areas? They are wider, uh, meaning more dense than bone. That's just how dense these cuticle is, these tissue actually forms. Okay. Now these tissues, when they have bacteria inside, they are not actually infectious. However, we know lung works hard, right? You breathe in, breathe out, meaning your tissue is you constantly work the tissue, right? And so eventually, if you have them long enough, these cubics inside will actually burst. These tissues will burst, and then the liquid inside will come out. And we know how bad that will be if you come out and then you're going to cough. When liquid gets into the your body reacts trying to get the liquid out. And so you're going to cough. As soon as you cough, when you cough, the bacteria just comes out. And if you happen to have the next person next to you, yes, they will breathe in, they'll get infected. And if these bacteria landed on the surface, it's not sterilized, and next person touches it, and they will get infected. All right, so that's tuberculosis. And so mostly this has been controlled, but in some special areas that it's still problematic. So here are structures of drugs that can block mycolic acid synthesis. So if you blocked mycolic acid synthesis, then the bacteria is much more sensitive to the antibiotic treatment. Even with this, the bacteria grows very slow. And so you need 6 months of treatment of antibiotic to control the infection. And because you have to take the antibiotics 6 months in a row, every day, every single day at the same time, right? And so that's a long treatment. Now, so if you don't follow the treatment, then the bacteria will come back and then cause an infection. And so right now, these tuberculosis cases are often arise more frequently in, anyone want to guess what population? It takes takes place often in prison population. Okay, in part because it's very densely packed area, and also people in prison are not the ones who will follow the regimen of taking medicines, right? They don't follow the law. They don't follow the rules, and so that's partially contributing to the spread of tuberculosis. So now the next I wanted to talk about cold and influenza and COVID-19, and that's all that is among the regular population that we should be concerned. So here is a breakdown or a tracking of about distribution of different infectious disease. So all microbial infectious disease and cold and influenza, meaning flu, right? So we're going to talk more about cold first. The cold is caused by what's called the rhinovirus and adenovirus, and cold can be caused by a bunch of different viruses, right? And so And if you heard about Johnson and Johnson's vaccine, it uses a adenovirus as the vector to, uh, and that's one that can cause cold, obviously re-engineered, right? And so the cold virus, they look like this is rhinovirus particle. They look like this. And if you look at this, it's not very clear, right? But at least one thing that you can come up with by looking at this picture about this virus is each viral particle looks pretty similar, right? And that is actually true. The viral particle, they very almost identical, okay? And so there are each strain of rhinovirus is the same. Uh, they reproduce the same and same proteins on the surface. So altogether, supposedly, there are 139 cold virus, all kinds of cold virus, but altogether there's 139. That means what? If there's 139 cold virus, that means you will have to, if there's no drug to treat it, then if you had 139 cold virus infection, not at the same time, hopefully, would you get another infection by cold virus? So last week we talked about the vaccine and vaccination, right? So if the cold virus is stable, and if you've seen it once, when you see it the second time, are you immune or not immune to the cold virus if it doesn't change? And you want to type in the chat. So the question was, rhinovirus doesn't change its surface. And if you expose it to once, and what happens and you got sick was cold, right? When you see it. But when you expose to the same rhinovirus the second time, will you catch cold? Anyone want to type in? Right. How many people would say no? How many people would say yes? I hope majority of you will say no because you are effectively vaccinated in the first exposure. And that means if you get 139 cold, you can keep a scoreboard. And if you get 139 cold, then you are effectively completely vaccinated while all cold, right? Now, so one of the things getting old, one of the good thing about getting old is that less likely you will get cold because you would have seen all the cold virus in the area. Now, just assume you don't travel, right? Okay. So that's about the cold virus, and there are some experimental drugs that can block or block the reproduction of the cold virus, but that's in the development. But most people simply just tough it out, right? Now, influenza is very different. That's the one that causes flu. And it has three different types, at least three different types, and that we know for now. And then what's also interesting is that it has 8 fragments of single-strand RNA as a genome. So we already know the single-strand RNA. Well, when it is functions a genome, it needs to be replicated or made into double strand and then copy the other side, right? That means it relies on RNA polymerase to replicate the genome. Now, RNA polymerase is not accurate. We know that. And it does not double check what it puts in, right? So it's more error prone. And so that means that have many consequences. That means it will mutate a lot, right? It will introduce mutation very frequently. So that's a unique feature about RNA virus that reminds you about the COVID-19, which is one long piece of single-strand RNA. And so this introducing a lot more mutations. So now the question is, we know mutation is bad, and we often use, when we later on we'll talk about AIDS virus as a treatment, we purposely introduce mutations so that it will be mutated so badly that the proteins are no longer functional. But why would this flu virus still thrive with such inaccurate genome replication? Anyone want to guess? It's had to do with the numbers of virus. All right, it's the population. And so, yes, there will be lots of mutations, but the more mutations that you have, the more variations you have, the more as a species, you'll be able to adapt into new environment and thrive. Okay. So that's the comments related, bring you back about everything we know about RNA and RNA virus. Now, this number of 8 fragment, and that's actually a very problematic for us. That means the virus have 8, each viral particle need to get 8 different RNA pieces into its genome. Okay. Now, it's a problem for the virus, but they figured it out how they can replicate and then sort it. Now, the problem comes in is if you have bird flu and human flu in one person. Okay, so right now, bird flu infects bird and causes flu in bird and kills bird, maybe cause a problem. And human flu circulates in humans. We more or less have gotten used to it. We can get vaccines. And however, the biggest worry is that what happens if the bird flu gets into human? So meaning if a bird flu infects one person and this person gets also infected by human flu. So now this one person will have 2 flu virus in its body. Each have their own sets of 8. Now, obviously, these can be mixed, right? So this 8 fragment of genome 1, 2, 3, 4A, 5A, and if you can imagine that you can have a bird and human flu hybrid, and that is actually could cause a major problem. I mean, worse than the COVID-19 that we are seeing now, because it will be completely new to us, right? And so that actually is one major problems that we're facing. Now, there's when we think about the flu, one thing on its surface has the antigen on its surface, and the antigen can drift, small changes, and shift, well, major changes. Okay, so those are concepts that we have to deal with. And so if we think about the flu virus, and this is what's on its surface, right? And so the flu virus is often designated as N1H1 or N5H5. What does N stands for? The N is this. Nuclear protein is N. H is hemagglutinin protein, it's this. So these are the two spikes proteins on the flu virus surface. Okay, so the flu virus actually has two spikes versus the COVID-19 that we know a bit more now has one spikes. And so these protein can change, and that's why they're characterized as N1 or N5. And so so the changes needs to be accounted for, and the vaccine needs to be different. And so each year the scientists will estimate, the epidemiologists will calculate the incidence of the flu virus that was prevalent last year, and or maybe it's not most popular, but it's on the rise, and so they have to calculate and project what flu virus will be dominant this year, and then they incorporate those N and H variants into consideration and make the vaccine that will target. Now, that's also the reason each year you should be vaccinated, because each year they are slightly different. And so if you stop vaccination, then soon, in a few years, your body will no longer recognize or have antibodies that can bind to H or N protein. And so that's the importance, right? Okay. And so before I move on, and I wanted to highlight the key difference between the cold and influenza. Well, it's different virus, but the symptoms are also different. The cold, you will have sore throat, runny nose, but no fever and headache. That's flu or influenza. In this particular year, I have to add another example of COVID-19, and hopefully that will we can rely on more we know by now. And so this is a graph that was projected last year. These are in 2020. And this projection actually turned out to be quite accurate. And so these are, remember, these are all the declarations or the major events. So Cuomo issues, stay-at-home order. Everything goes up now. And the first case is here in New Rochelle. That was the first case. And first death. And then it went up. And daily record of 12,000. Okay, and then face mask, and so these are by June, July, and that was mostly quieted down. Okay, actually, this curve was predicted by epidemiologists very early in March, and I was one of the people that was remain optimistic, very optimistic, thinking, okay, by June, July, we'll be okay, we're back normal, and we'll go back on campus and start teaching on campus. and start doing research on campus. Remember, I have a research lab, right? NIH-funded research. Well, we will all block out of the campus. We can't do any research during this period. Um, Now we know what happened after that. We had another peak, and right now, for this fall, you read in the news that we will come back to, we're going back to campus for in-person class. But hopefully, the rules are still murky at this point. The governor says we all need to go back, but there's lots of questions. So, but what I wanted to spend time here, though, is to talk about the COVID-19 and why, what does it do? So this is a picture basically showing you about the COVID-19, the life cycles of COVID-19. So the COVID-19 virus binds to the surface receptor called ACE2, and it gets into the cell, our cell, whatever cell it gets in, and it in going through endocytosis, meaning it has a membrane. However, this membrane will break down, and then the mRNA come out, RNA will come out, and then starts to going through the life cycle, incorporating RNAs, and eventually make the viral particle. Okay, and then come out and infect other person. Now, at the same time, the infection will can cause these the cell that's infected will produce interferon, and that will reacting, and to get the next cell start to have proteins or to inhibit the viral particle. You see, all these are being produced, blocking. So our body, the now these are possible treatments that can be that we can have, but I think I would just highlight what I think is important is here on this particular slide. So the interferon is being produced by the infected cell that allow the neighboring cell to be ready to alert the neighbor cells to stop preventing the infection. So that's that's what we have. Our cell naturally have that ability to control, okay, infection. And in the next on this slide, we'll need to focus on is this binding. The AC2 is the receptor. Okay, this S protein. So this S protein binds to the ACE2 receptor. Now this receptor is on many different cell surface, a lot of them. And so the binding then allows the infection. And this can be the enzyme can activate the S protein and make it more effective in binding the ACE2 receptor. Okay, so now the main focus is on this the S protein on its surface. So this virus, the COVID-19, this is the viral particle. Okay, and it's lined with spike protein, which we call S protein. And the S protein has this binding domain. Okay, this is the part that binds to the ACE2. Okay, and so this is the part that we are using. The S protein is the part that we put them use mRNA to code the S protein, and then use that so that the now you understand the the S protein is you make an mRNA that's the S protein, and then you wrap them up into a lipid nanoparticle, and that's what's being injected into your body, and in vaccine, and then that lipid nanoparticle will get into bring the mRNA into your body, and your body start producing the mRNA. And so this is the vaccine strategy. How does it work? And so this is your needle with the nanoparticle with mRNA inside, and that delivers the mRNA into the body. And now you start producing these spike proteins. Okay, so the spike protein is being produced, and then once the spike protein is produced, then it can be either taken up by macrophage or taken up by the B cells. And so now then these B cells and into, remember, the B cells, and then you have the into the B cell be activated, and then you have long-term memory cells, and then plasma cells, and to produce antibodies, right? So the antibodies will have variable regions that can bind to these spike protein, and the spike protein then it will be on the surface of the virus, right? And so the spike protein that you the antibodies that recognize a spike protein that can bind to here. Okay. And so that will prevent the infection. Okay, so this is a schematic, really. I made it up to show you. So if you have the virus, and if you want to stop virus, you want the antibody. And the antibody can be made in multiple different ways. Traditionally, you just take this virus and breaks up into pieces and then inject into the body. So that's one vaccine. And then your body reacts to this vaccine, produces antibody, then that's one. And the new way, more effectively, is to use this approach, the mRNA approach, right? And then you have now antibody recognize the virus, and that prevents the virus from getting interact. So if you have spike protein all covered with antibody, then the spike protein cannot interact with ACE2, right? So now this virus is really blocked. Now, recognizing the role of your antibody, and wouldn't it be nice if you simply produce lots of antibodies that can simply block this? That's the approach taken by Regeneron. Remember, Trump was when Trump was infected, he got treated with a Regeneron antibody treatment. They gave him 8 or 9 milligrams of antibodies. That's a huge, huge amount. Basically, you flood the person's body of blood with antibodies that recognize the flu virus, the COVID-19. So now the COVID-19 virus is essentially completely covered by the antibody, and then this virus can no longer replicate, and then you basically kill the virus. You stop the virus from being infected, right? Infectious, and so that's a very effective treatment, right? Except that this thing is hugely expensive, so they don't use it for also It only works when you have early detection. It only works when you have small amount of virus. If you have too much late stage, it's proven, not proven, it seems to show that late stage people get infection, and this doesn't help because the virus is already in the tissue and start destroying your lung tissue. And they go from tissue cell to tissue cell. They don't go through the blood. Now that's when this doesn't help as much. Okay. So anyone have any questions about COVID and flu and um, vaccine? Any questions? Okay, so you all are on the front line of explaining to your friends and families how vaccine work. I have, we should have the um, this class being on the forefront of explaining. You guys are the best, most educated person on campus about the COVID and how it works and how to treat, right? Okay. All right. So the next thing is about a few examples of direct contact bacteria can cause infection. One of them is Helicobacter pylori. It causes gastric ulcer. And this, and you guys were all born very fortunate after we figured this out. Um, ulcer used to be a very, very common uh, disease, not disease, common, yes, disease, common problem. And you have to get treated, and sometimes it gets so bad they actually have to do surgery because your stomach gets so damaged, bleeding, and it is really a problem. And but then there was a doctor in Australia and who got interested in this, and he's a surgeon. He cuts out these tissues, and then he collaborates with a doctor, and they together they found actually there's a close association with this helical pylori. And so well, initially nobody believed them, and they all say, well, yes, if you have a wound, obviously you're going to have bacteria colonizing the wound. The question they were saying is, well, actually, the colonization of this bacteria is the one that causes the wound, and then eventually lead to the ulcer. So what actually later on what's found out was, well, this bacteria, well, we know stomach acid is pH. Anyone know? How acidic is your stomach acid on pH scale? Someone must know. pH what? pH 12? I'm gonna play my daughter's guessing game. pH 12, 10, 8, 6, 4, 2. Yes, thank you. pH 2. So it's very acidic. And so we already know bacteria, when it gets to so low, they can't even grow. And so then they will have to, so this bacteria, so almost all bacteria will not grow in this pH, then they die. But this bacteria found a way to colonize the fold of your stomach tissue. And then in that fold, they produce ammonia to neutralize. So in that little crevice, they basically found a cave in your stomach lining, right? And then they will grow in the cave, and then they will flood their own little cave with ammonia. So the pH in that little environment is actually 5, and so the bacteria can grow happily. And well, you provide lots of food, so they're never short of the nutrient, and they just need to create this little environment and they can grow. And so, as it turned out, you get colonized very early on by um, the helicobacter of your mother, um, and because your mother feeds you, and her helicobacter will colonize the baby. Now, it's probably very easy at the beginning because baby's stomach is probably not that acidic. They are not producing that much acid, right? And so, but as we grow older and these bacteria get established, and when you get to 40, uh, they're there too long, and then they start really causing problem, and they bury themselves so deep, and they grow so much, they break down the stomach lining, and then your tissue is exposed, and your tissue is not resistant to low pH, right? Stomach acid, and that's when you have stomach ulcer and continuously. And so once we understood that, and once we gave those doctors actually received Nobel Prize for recognizing that problem. And so so now we actually can treat them easily by providing antibiotic. Okay. There's another set of very difficult viral disease is hepatitis. Hepatitis, there are at least 6 different types of hepatitis, and there's a vaccine for A and B. And if it's untreated, the type B and C can cause chronic infection in the liver and eventually lead to cancer. Okay, and the part of the problem is these hepatitis virus can go through in other organisms, and then they're not causing disease in other organisms. We call them viral carriers, like the sea clams, and they get colonized, and the virus will just grow, but doesn't cause any problem. But when we eat those contaminated, then the virus get into our body, and then they can cause a viral infection. So scientists are really working hard trying to get a vaccine for all types of the virus, hepatitis virus. And I remember when I was in college, one of the hot topics was to get one of our lab, not the lab professors, was working on getting hepatitis C vaccine. Okay. And that was really difficult. But I'm hoping with the mRNA, the new mRNA viral vaccine, maybe that will give us hope to create new vaccines that it was impossible to create in the past. So here are the infection incidents of different hepatitis virus, up and down. And the C is low, but both ABC are types are coming down. It was really bad at the beginning, well, late in the 1975, and that range was very prevalent. And the vaccine, introducing the vaccine B and A, and then you see both incidents coming down. So the vaccine does work. And so here's a picture of the hepatitis B, HBV virus. So now I wanted to spend some time talk about the sexually transmitted microbial infection disease. And I will talk about gonorrhea and chlamydia and HIV. Gonorrhea is caused by Neisseria gonorrhea. It's a gram-negative aerobic bacteria. It's fortunate for us is that it's very sensitive to drying. Now, if you see, I don't know if you were shown pictures of this bacteria. And this bacteria, it's very, very small. And that's one. Two is it always, you can find them always, two of them together, right next to each other. Almost like always existing timer, two bacteria cells side by side. And we often stain them, and you can see them in the pink color, very tiny. And one of the problem that that causes continuous spread is that the infection in female is not obvious. And in terms of treatment, that is actually antibiotic treatment still works. We can use antibiotics. And so here is incidence of gonorrhea. And so it's historically it fluctuates and it goes up in 1945 and World War II. And syphilis is World War II related, very high up. And with the birth control, the introduction of birth control, it got a high incidence peak, and it finally come down. People recognize that the safety is important. So then protection is put in, and so now the incidence is coming down. And one of the reason is that it's a specific epithelial cell that in the vaginal tunnel has these cells, and so these bacteria then attach to these specific epithelial cell. It's always there. You see two of them together. It's two and two. It's very strange. They always do that. Even here. So this is two, and they're probably two. And also these epithelial cells are also in your eye as well. Okay. So now it becomes a standard practice when baby is born through vaginal delivery. And the first thing the nurse do is simply just wipe the antibiotic ornament onto baby's eye. And so that's why you see babies always have shining when they first come out, and from the delivery room, their eyes are shining with that something on it, and that's the antibiotic ornament. And that's what the nurses are putting them on. And it's unfortunate that the penicillinase producing gonorrhea is on the rise and it keeps going up. And so remember, this is the enzyme, or they can be called beta lactamase. And this is the enzyme that breaks up the penicillin ring that's consistent, uh, that's conserved for all enzyme or all antibiotic in the penicillin family. So ampicillin, amoxicillin, or any of these cillins, um, they all have that conserved ring, and they are then sensitive to uh, uh, so these enzymes, they can uh, they cannot stop this bacteria, okay, because beta penicillinase was break down all the penicillin, amoxicillin, and allow this bacteria to grow. Okay, so that's a real, fundamentally, that's a real big problem for us, the rise of antibiotic resistant bacteria. The next bacteria we wanted to talk about is chlamydia, and this is a very unique bacteria. This is Chlamydia trichomonas. And this, when we think about bacteria, we always think bacteria um, if you gave them media and they would just grow in the media. Well, this is a parasitic bacteria, and it will grow, but what it will grow is it will infect human cells and get themselves into human cells and then grow inside the human cell. Right? And the net result, so they got more specifically, they will go to the microvilli. They have special receptors, allow them to attach to microvilli in the fallopian tube. Okay. Now, and then they will grow inside, and then they will kill this infected cell. The cell will die. And so now this becomes a lesion, right? You see this here and here, these cells are infected and killed. And now so now your body reacts trying to heal the lesion. So remember, if you have a cut in your hand, you can always see the scar, right? The hand will heal, but you will have a scar. Now imagine that scar is in the fallopian tube. And so if you have repeated infection of this bacteria, and the tube will essentially get blocked, and then women become infertile. Okay. Now the treatment, so the infection is not apparent because you don't have nerve ending in the tube, and so infection is not detected by your body. The infection can be detected by ELISA assay. So meaning that if you have infection, your body have this bacteria, you will have antibodies against this bacteria, which can be detected by ELISA assay. Now, let me quickly ask, is any question about ELISA assay? Okay. Now, in terms of treatment, penicillin is ineffective. Okay. This bacteria, they don't grow outside, they grow inside the human cell, where penicillin is less accessible. Okay. What is accessible, what can be treated is deoxycycline. And that is the antibiotic that can be used to treat this chlamydia infection. Right? And so people do need to periodically get checked to see if you carry this bacteria, because unless you look for it, and you wouldn't see it. And so the doctors probably do routine annual exams, probably do check. And so this is uh, the antibiotic, deoxycycline. So the next, what I wanted to talk about is the HIV infection. And throughout the semester, we talked about different aspects that can be brought on to understand this infection. All right. So let me go through. So first, the disease is caused by the HIV virus. It binds to the surface and then delivers the viral particle inside the human body. And this binding itself is targeting the, so now we know the binding a bit more now. So the viral particle has this protein called GP120, and the GP120 is targeting the CCR5 and CD4. It's stabilized by CCR5 and CD4. CCR5 is a protein that on the surface of most our cell. And so now this, and then CD4, remember CD4 is a T helper cell, right? And the T helper, so now the HIV virus is targeting the CD4 T helper cell. And that's why then if all your CD4 cells are killed, the immune response will not be activated, right? So you don't have the macrophage activated, you don't have B cell activated. So now you wonder if you can, what does CCR5 do? Can you simply get rid of the CCR5? Well, there are some evidence suggesting that CCR5, you get rid of CCR5, then you are immune to HIV. Supposedly there is a population in Africa reported that they don't have the CCR5, and they are immune to HIV infection. Okay, so that's the critical part of the infection. And so these are the viral particles. So these are the mature form that's coming out. And so this is actually showing you the viral particle that's coming out of a infected cell. And so that's the problem. The cells are continuously produce these viral particles, right? Because we know the viral particle goes into the cell and then reverse transcribe and then integrate into your genome. And then your genomes is keep producing these proteins, viral RNAs, and then they are being packaged continuously, and then you continuously produce these particles. And these then go on to infect another CD4 T helper cells. And that is a continuous cycle. So if you were to track the CD4 cell, this is what we'll see. So initially, so the dashed line is tracking the virus, viral copies, if you track viral copies. And this is tracking the CD4. And so when you initially have an infection, you have lots of virus, but then the virus actually gets contained a little bit, and then because your CD4 cells start kicking in, T cells start kicking in, trying to control it. But then it starts to killing the CD4 cells, and you then start produce more CD4. You right because the CD4 will to to uh, to kill to control the virus. But so then the viral particle maintained the same level. However, this is actually the result of the fact that you continue to produce the CD4 cell, but then you're producing so much CD4 cell is very, very taxing, meaning tiring. And you are actually will not be able to keep up. Eventually, the virus will kill more cell than you can produce. And the CD4 continue to go down and go down. And then at this point, you can start to see secondary infection start coming in. And eventually, the secondary infection are the ones that kills the people. And so normally you wouldn't have these fungal infection because your body is able easily kill these fungal infection, but those fungal infection can go on and then spread to your body and eventually kill you. Okay, and there are some treatment. So at this point, there are some different categories of drugs that can be used for treating to contain the virus. So right now, these drugs are pretty effectively blocking the virus from mass produce. So you can keep the viral low at very, very low level. So then your body will continue to have enough CD4 cell to control the secondary infection. And so I think some of the patients has been around for 20 years now. So the HIV infection nowadays is a manageable infection. It's no longer a death sentence because in the 80s, if you have it, you die. Now it's more of a manageable disease. Of course, we're trying to find additional ways to prevent the HIV from infecting. And so, for example, you can have these are the new strategies. So we know CD4 and CCR5 or CRC4 are the receptors, right? And so what then you can do is you can produce GP120 inhibitors, or you can have inhibitors or proteins, something binds to CCR5 and block these interaction or this binding. Okay. So in summary, we have the HIV virus that can cause by HIV-1 more virulent version than HIV-2. We know throughout the semester about the structure of the virus and the life cycle of the virus. It incorporates into the genome, makes it very, very difficult to kill, to remove the virus after they got into your body. And then in terms of infection, it's targeting the CD4 cell, T helper cell, and it's also targeting the macrophage that has the CD4 and CCR5, and the multiple diagnose based on the symptoms or on the virus or on the antibody. And the treatment is you have protease treatment, and you have those three different categories of treatment that we can actually keep the infection in control. But unfortunately, at this point, we haven't been able to completely cure the infection. You've heard incidents or reports of a few people gets cured of HIV, but then they stop treating them, and eventually, after a while, the virus become detectable again. So meaning the treatment, we can treat them, keep the virus low, because as long as you keep the virus at very, very low level, your body is able to produce enough CD4 to prevent a secondary infection, and then you're fine. You can live for a long time normally. Okay, so that's the treat. So those are, so this slide summarizes all the major topics, all the microbes that we talked about. And if you're thinking about review sheet, this will be the first thing you want to get to.